HDF have been pivotal in Huntington's disease science research families since the very inception with Milton Wexler.
Organizations like HDF play a critical role in science. The kinds of collaborations that emerge, the new ideas that come out of it, it goes far beyond any individual funding of an experiment and more about creating that sense of community.
They support the research of scientists all around the world.
HDF is amazing because they don't just provide funding, they also bring the community together to discuss ideas and inform each other's work.
You feel like the hope for a cure is closer when scientists are all in the same room.
We need support systems for HD patients, we need funding for HD researchers, and we need a place to come together to talk about advances and for researchers to understand what's going on in the HD community and vice versa.
The hallmark of the HDF now is its brilliant ability to attract and support younger researchers, keep them in the field long enough for that to become their life's work, and then to connect them to other scientists who can help them to accomplish their goals.
Welcome, everyone. Tonight, we focus our annual gala on what's next in Huntington's disease research. We look forward to updating you on many exciting topics from some of the leading minds in Huntington's disease research, and doing so with a strong sense of community for which the Hereditary Disease Foundation is known, and which each of you help provide. Thank you. Tonight's topics are varied, and the backgrounds of our presenters are diverse. But what will sound very familiar to you is a strong sense of community and collaboration, which have been the defining characteristics of the Hereditary Disease Foundation, galvanized by our president, Dr. Nancy Wexler, since our founding over 50 years ago. Tonight, we will meet the global community of researchers whose work we fund, working in countries around the world. We will also visit the laboratories of several of our scientific advisory board members. I would like to thank each of our Scientific Advisory Board members for their steadfast commitment to pursuing the work needed to develop treatments and cures for Huntington's disease. We are pleased tonight to celebrate the accomplishments of Dr. Natalia Barbosa and Dr. Stephen Finkbeiner. Dr. Barbosa has been awarded the Nancy S. Wexler Young Investigator Prize for her stunning work that has the potential to change the way we think about Huntington's disease and present new ways to develop treatments. Dr. Steve Finkbeiner has been awarded the 2022 Leslie Gary Prize for Innovation in Science for, among other things, the creation of the robotic microscope and his work in artificial intelligence, which have made huge impacts on neurodegenerative research. Finally, we will celebrate the work of the Hereditary Disease Foundation's Young Investigators. With your support, the HDF has always invested in the careers of promising young scientists by providing much-needed funding for research that furthers the HDF's mission. It is this support that helps keep these young researchers engaged in the fight against HD long-term, building on the work of their mentors. These young investigators inject new energy and ideas into developing treatments and cures for HD, and we are thrilled to share their exciting stories with you. As always, however, our gala, the journey of our young investigators, the successes of our leading researchers, and the hope that our work provides to HD families around the world could not be accomplished without your generous financial support. We are grateful for your participation in our gala tonight, and I hope you will join me in making a meaningful donation online to fund all of the great research about which we will now hear. Thank you in advance for your generosity. Now, let's meet some of the Hereditary Disease Foundation's extraordinary researchers.
HDF attracts young scientists like me to embark on Huntington's disease research. People like me who were not even aware of the disease a few years ago, now, because of this support, some of us like me are going to center our future work on Huntington's disease.
I think young researchers bring a fresh set of eyes to old problems. We can look back at old data and old established truths and kind of re-evaluate how real those are. And I think that's a really important role that young researchers can have within the HD research community.
Research is basically how we're going to evolve as a human species.
My work will make a difference because it's a piece of the puzzle, the big puzzle of finding a treatment for HD.
It's very motivating and powerful that we as scientists can transform the I don't know into I don't know yet.
What drives me is not curiosity alone, but also a small purpose that I can contribute to find a cure for this disease.
You have to keep your eyes on the prize. How is this going to lead to a treatment for HD?
My work will make a difference.
My work will make a difference.
My work will make a difference.
There haven't been any drugs that have been able to stop or cure a neurodegenerative disease. But where we have power in Huntington's disease is that compared to something like Alzheimer's or Parkinson's, we really know what the mutation is that causes the disease, and it's the same for all patients. We're really interested in developing small molecules which can pass the blood-brain barrier and directly bind to the DNA mutation. And so what I'm doing in lab is I'm characterizing one small molecule that we've identified, which binds to the repeat expansion that causes Huntington's disease, and actually is able to reverse that expansion. So it makes the repeat tract smaller in size. And so we can move from a repeat expansion to something that looks more like what you'd see in an unaffected individual.
I've always been interested in understanding that level of detail about human disease. What is the very beginning point of a disease? Where does it all begin at this very molecular level is what really interests me. So rather than looking at a whole person with Huntington's disease, we just look at the single Huntington protein molecule. What we try to do in the lab is we will make that protein and then we can use it in all sorts of different experiments to try and understand what does the Huntington protein do? What is its precise molecular function and how is this changed when you have the HD mutation? Rather than just looking at sort of big, broad strokes, abstract ways, we can really pinpoint what's going on.
Urgency for me is very much stems by the fact that there's over 30-something people at risk in my own family. For some people like me, knowledge is power and you want to know everything and do everything. My current research has been working on what we call biofluid biomarkers. We know people carry the gene and we know they will get symptoms at some point with Huntington's disease. Biomarkers could allow us to take snapshots of people in that in-between stage and to track the disease a bit more closely. What's particularly exciting is that it appears to change very early on in the course of disease. We could potentially use this biomarker to run trials and have a way to measure if a drug is working, which I believe is going to be the way forward in preventing HD from ever happening.
We found out when I was finishing graduate school that my mom has the Huntington's gene. And once we knew that mom had the HD gene, I just wasn't interested in working on anything else. So that's what got me interested in the HD field. I am absolutely on a mission to help people with Huntington's disease. DNA is what makes up our genes. And our genes, they have to be in good shape for our cells to work properly, but it gets damaged all the time. So DNA repair is really important. Our cells have evolved many ways of repairing the damage, but there are neurodegenerative diseases caused by DNA repair that's not happening properly. And when those things go wrong, that's when you have disease. There's this one ingredient in the DNA repair process. It's called poly-ADP ribose, but we call it PAR for short. And PAR gets added to sites of DNA damage, and it kind of acts like a fishing net to pull in all of the proteins that are in charge of repairing the damage. PAR has been implicated in neurodegenerative diseases. So we wanted to ask, is there a problem with PAR in Huntington's? And as it turns out, there is a problem. We actually found that the Huntington protein physically binds to PAR. I think that our work might contribute to a conceptual leap in HD research. I'm on the clock for possibly myself and many loved ones.
If I were to take you into my lab right now and show you the most exciting thing that's there right now, it wouldn't be a particular experiment, it wouldn't be a particular project, it would be the people.
There's been energy. We have so many enthusiastic, younger researchers, and that's what's going to keep driving things forward. We need to have a healthy research landscape in order for us to succeed as HD researchers.
Was there a specific thing you wanted us to look for?
Collaboration is fundamental. To solve a problem like Huntington is incredibly hard. You need to know about neuronal physiology, biophysics, biochemistry, pharmaceutics and development. So it's very hard for one person to know all of those things well enough to be at the cutting edge of what is needed.
It's important for mentors such as myself to instill that sense of collaboration amongst young investigators, to help them understand their place in the scientific community and how to collaborate effectively.
You can't be a one-person scientist anymore. I couldn't do this if I didn't have all the collaborators I have. I have also started collaborations with other HDF fellows, and we've been trying to understand different antibodies that are available for the Huntington protein.
Open science is really how you're going to get science done faster and better. And I think if we want to find a cure for Huntington's disease, you have to be open and you have to be collaborative and you have to be willing to share. Otherwise, it's going to take us much, much longer to get the job done.
What's really exciting for me right now in Huntington's research is we're expanding beyond our traditional sense of what's actually causing this disease.
I feel like the HDF is one of the best organizations which motivates the young investigators.
The HDF have been really important for supporting my career. I felt really supported as an individual and really valued as a scientific mind that they want to keep in the field.
Supporting young scientists from early on is key to attract more and more researchers into the field.
If we don't fund people at the beginning, they're not going to be there. We're not going to have the experts at the end.
When I was told that I received the Nancy Wexler Young Investigator Prize, I couldn't believe. That was very exciting news to me. I was grateful for the recognition I was receiving from all the years I've been dedicating to science, and I also felt a strong calling to push my research to next level.
For HDF families and supporters, there is hope.
Now is a time for hope. This is the time to continue to move along with the scientific advance that is part of the hope, which is actually getting stronger towards being able to treat and or cure Huntington's disease.
We all want to go and find a cure, so we have all common goal.
You always get the question like, how long is it going to be? And I don't know, no one knows. And my only answer for that is we are working as fast as we can. Working as fast as we can.
When a foundation recognizes that your science is actually impactful and meaningful and that it's important, it shows you, oh, I can make it there one day. You feel like you want to go into the lab and discover more things.
My work will make a difference.
My work will make a difference. My work will make a difference.
One of the things that we try to do is really think first, what is the ultimate goal? Where are we trying to get to? And then take a step back and think about, what do we need to get there? In some cases, the approaches that already exist are sufficient, but in other cases, it's pretty clear that the major hurdle to getting where we need to go is the unavailability of approaches. And so in those cases, we try to think about, can we invent a new approach that would be able to enable us to get to where we want to go? Huntington's disease was the first disease we really focused on, and it was, in fact, some of the unanswered questions in Huntington's disease that drove us to invent technologies to try to address them. It's about really making a difference and trying to impact this disease and bring a cure to patients. The Center for Systems and Therapeutics really began with trying to focus on the question we were trying to solve, which was this question of, in Huntington's disease, we were initially narrowly focused on what role those protein deposits play in cells. But I think as we started to think about that problem, we realized that it's just one example of a recurring question that comes up in many diseases, including Huntington's disease. You could do a lot of possible things. So you could create and build maybe instruments that did things nobody done before. And if the path to get there doesn't exist, let's invent it.
What we see is that some individuals get HD really early on in their 20s, whereas others are able to stave it off, up into their 70s. And so the question that I'm really interested in understanding is, why is that? What changes in DNA are different across individuals with Huntington's disease? So we know the causative mutation that gives rise to Huntington's disease. It's in the Huntington gene. It's absolutely certainly must be the case that other genes play a role in how the disease unfolds. And so we're searching for variants or changes in other genes that may play a role. We've done a technique called whole genome sequencing, which essentially allows us to understand every base pair across the human's entire genome. We've recruited many participants with Huntington's disease, and we've collected patient data, disease trajectories, and also age of onset. We sequence their DNA, and then we're using bioinformatic approaches to try to understand essentially what's similar across individuals that may have more resilience to the onset of HD or perhaps are more susceptible to earlier age of onset of HD. And we're trying to find if there are particular variants in genes. But it turns out there's millions of variants. And so how do you find out which variants are actually really, really important? Machine learning is being able to identify signals in cells that really no other approach has been able to do, or certainly the human eye. It's way beyond the pale of what the human brain can see. You can't see a difference in the cells, but these algorithms are able to. That opens the platform for understanding the pathology. And if we understand what the genes are, those may be particularly important, possibly genes that we can target therapeutically. There's a lot of possibility from harnessing technologies that I think we never had before.
I'm a biologist trying to step in the world of computer science. Machine learning, deep learning is going to be the future of biology, where we can do a lot more things that we can right now. We want to see how we can build technologies that shape the future of research. We're using machine learning that has been used outside of biology and try to bring in and apply to the biology question that we're asking. We use tools that might be using in auto-driving cars to recognize surrounding features. And we are trying to use similar approaches to train machine learning models to recognize cell movement, to recognize patterns, to quickly identify the cell populations that are more vulnerable. And as a young scientist, we are very interested in how do we build tools to solve the questions that are still unsolved over the past years. And then we want to see how we can build technologies that shape the future of research.
The main technologies that make a difference for us in the field is being able to use robotic microscopy and combine it with deep learning. So with robotic microscopy, we can image a lot of cells, many, many more than you can do on a regular conventional microscope. And with deep learning, we can analyze all those cells instead of just analyzing a couple of them or a handful. What we're trying to do now with hyperspectral robotic microscopy is typically we can look at a cell and we can see it die off. But to understand why that cell is dying, we need to also be able to visualize other parts of the cell. We need to be able to see inside the cell to see what the mitochondria are doing, which is the energy house of the cell, what's the cell nucleus, how that is changing, or the DNA is changing. We're hoping to build a drug discovery platform where we will use deep learning to identify if a cell looks healthy or diseased. Then we can use that deep learning to try and change the fate of the cell. We can apply drugs to the cells and then see, will the cell change from looking like an HD cell to looking like a control cell? What researchers are trying to do is to keep cells alive instead of having them die off in the brains of patients with HD. It's really our goal to get the disease treated or even cured and make a difference.
I care deeply about making a difference, and it's in my bones. And so I feel like one of my roles is to be able to not only articulate a goal, but to get people excited about it and to rally people around it. I've really tried hard to do with my group is to never feel limited by the tools or technologies that are available. So have a clear idea of the question you're trying to answer and the goal you're trying to achieve. I'm thrilled by the prize and deeply grateful to the foundation. But the prize is incredibly meaningful to me, partly because it's a prize for innovation. One of the really brilliant things that HDF has done over the years is that it's tried to find promising young scientists to give them support at an early and really vulnerable stage in their career. And most of us never forget that. We not only fall in love with Huntington's disease, but it becomes then a lifelong career, as it has for me. You finally can do something you've always wanted to do. Now you're starting to think, what else could I do, and what's the next big problem?
In the beginning, it's a scientific problem. It's an intellectual problem.
For me, what drives my research is curiosity, wanting to understand the unknown.
The way ideas normally start is either because they've formulated hypotheses tested based on literature or observations, or just serendipity, luck.
The families, the challenges, the patients, it's a fight. It's a real struggle. And so the idea that I could be able to work, dedicate my life to be able to really help, hopefully unravel and understand the causes of some of these neurodegenerative disorders. That's what's inspired me.
If you did a landscape of opinions across the HD research community, a lot of them would say, yeah, well, we're trying to heal our friends. What I'm really trying to do is make a translational difference in a clinic to help someone with this disease. This is definitely the golden age of HD research. If you think about the number of papers that are being published, the number of people that are now migrating to the field of HD research, the amount of resources, the number of companies like mine that are finding this a desirable space to interrogate for disease-modifying therapies, it's unprecedented, and we've never seen it before, and so it's absolutely kind of the golden age.
I'm very excited, having been in this field for a while. On the therapeutic side, I think there's been a lot of exciting developments of new approaches. We understand probably the system well enough to be able to design things that are going to work in terms of interventions.
The Structural Genomics Consortium is a consortium of pharmaceutical companies and academic researchers that are seeking to accelerate and facilitate drug discovery. We address all the human proteins and understand how they work and figure out which ones are drug targets and which ones can be modulated for therapeutic benefit. It's a really versatile platform that forms the basis of early stage drug discovery, where you make the protein of interest and you test what it does biochemically and try to figure out ways you want to go for a therapeutic intervention.
We started a translational center, initially narrowly focused on Huntington's disease, and it was really designed to try to cross that valley of death, to get drug companies to be interested enough to pick them up and to move them forward. And that was called the Toby Corrett Center for Neurodegenerative Disease Research.
We can take a blood sample from anybody and it can be reprogrammed, and they're able to revert them into what's called a pluripotent stem cell, meaning that cell can become any tissue type. It can become a neuron, it can become part of the stomach, it can become any cell type. And for us, for Huntington's disease, we really want to generate the neurons that are most affected in the disease. So the medium spiny neurons, the cortical neurons, we can then grow those. And so what's so transformative about that is bringing us the ability to study human disease in a laboratory setting directly from cells that come from patients. It puts us in a new space and time because we're now examining cells with the genetic background on all of the nuances. Now that we're much closer to the disease, we're much closer to the pathology, we're going from the variation in humans to validation in human cells in the lab, and I think that's really going to bridge the gap for our understanding, unlike any other time before, I think, in science, at least where we're at now.
That's where this translational step happens, where you have something where you feel like there's enough evidence to suggest that if you could develop a way to be able to target that in patients, that it might make a difference. We've done many, many collaborations over the years with large pharmaceutical companies, small biotech companies. If you have something that works in model systems, then oftentimes you can engage drug companies to be able to work on it together, or even to take it and to be able to develop it in the drug company into something that can be tested in patients and clinical trials.
And in the past 5 years, we've seen a tremendous amount of interest in HD. We still find this as a very attractive and doable disease area. We can make a difference in this disease, and I think that's recognized across the industry, and that's why you're seeing so many companies working in this space.
This is the first time in the history of Huntington's disease that we now have data coming from people in the clinic, in clinical trials. We're in the clinical trials era of Huntington's disease. This is new. This has only happened for the last few years. This was a dream 5 years ago. We have these fantastic clinical studies like Track HD, where we have 16,000 people or more enrolled. This is almost complete saturation of people who have the disease in the Western world. And we're watching every single one of them. And we've got all kinds of samples coming from them, and we're tracking them, and we're watching them over time, and we're watching them before they get sick, while they're sick. And all of this information is just a massive amount of data in real humans with real disease. That is an unheard of level of participation by a disease community. When we mention these numbers to drug companies, they're completely blown away. I have lost count of the drug companies that are interested in developing HD drugs.
I hope that the continued investment by pharmaceutical industries, as well as the increased work by the academic researchers, is providing a sense of hope to families. We will modify the therapies or modify our designs in our trials to continue to move the ball forward, and we won't stop until we succeed.
I think right now is a very exciting time.
Huntington's disease in the beginning is very much an invisible disease. I am sitting here today looking like a completely normal person, but I have a lot of my own battles going on behind the scenes. Having Huntington's feels like I have a very misunderstood disease. When people do know about it, they really don't understand it. It feels really lonely at times because the only people that really understand it are people that also have Huntington's disease or their families.
For the individuals who had a parent with Huntington's in their childhood who unfortunately had a lot of behavioral symptoms, anger, outbursts, irritability, even violence, the trauma is hard to fully work through and is never forgotten and affects the development and the life course of that individual. And we have opportunities to help people to work through that, but those wounds don't fully go away.
in the early 2000s. My dad started having psychotic episodes and his had personality changes. He wasn't going to work and he was becoming violent and angry. Not knowing what was wrong with my father, did a lot of research on the internet, and every time we put in what his symptoms were, Huntington's disease would pop up. We would kind of go, just to think that there's a 50% chance that each child will inherit the gene. It was quite scary. Right after my dad passed away, I decided to get tested, thinking I'm nothing like them. I'm not cranky, I'm not mean, there's no way that I have it. And I tested positive as well, which was a major shock to me because I was convinced that I didn't have it. Going on in my mind, there's depression, anxiety. I have a lot of trouble with short-term memory. I have issues with my balance, and I can't multitask. I started just forgetting details. I had a 30-year career in advertising and photo production. And 2016, I stopped working because I couldn't keep up with my job. When I tested positive, I said to myself, and this is kind of corny, but all right, I'm HD positive, and this positive means that I'm going to be positive about it, and I'm going to do positive things. I'm not going to make it a negative, I'm not going to let it weigh me down and being very negative. I'm really aware that I'm lucky to be this far along in the disease and not have as many symptoms as I do.
We can make huge differences in the quality of life for people in the premotor or early phases of Huntington's by treating emotional symptoms, psychological symptoms, creating a multidisciplinary team and a place of support, helping them plan for the future, and dispelling their overly catastrophic views or addressing their fears and their hopes.
I would love to think that I am an example to people that have Huntington's disease, to give them hope and show them that with doing the right things, with exercising, eating right, and seeing the right doctors, that you can stave off Huntington's disease for longer than you think possible. And there is research out there that's definitely something that helps me to stay positive. The research is really where it's at. That's what's gonna bring a cure to this horrible disease. And what I like about the Hereditary Disease Foundation is that their money goes towards research. When I think about the future, I hope that it's a future free of HD, but I hope at least for my daughter's generation that it'll be a world free of HD.
Denise's story is a vivid reminder of why it is critical for us at the Hereditary Disease Foundation to continue funding research to develop treatments and cures for Huntington's disease. We are grateful to Denise and her husband John for inviting us into their lives and engaging with us in a very personal, honest, and moving discussion of what it is like to live with HD and the impact that it has had on them and their family. Huntington's disease affects all members of a family. Those who have the disease and struggle with its symptoms, those who are at risk and live with the fear of one day developing the disease, and those caring for a loved one or many loved ones with HD. For the countless families around the world affected by Huntington's disease, for families like Denise's, like mine, the extraordinary work being done by the scientific community offers great hope. The researchers highlighted in this program and others across the globe are working to unlock the potential for new therapeutics and cures for Huntington's disease. Their research may also shed light on treatments for related brain diseases like Parkinson's, Alzheimer's, and ALS. The laboratories we visited introduced us to groundbreaking technologies such as artificial intelligence, machine learning, state-of-the-art imaging, and advanced robotic microscopes. These innovations are changing the way scientists study disease. We also learned about the important collaborative efforts taking place in the HD community. Through the global sharing of resources, expertise, data, and samples, researchers are better able to understand the disease and advance science faster and with greater efficiency. These advancements and shared efforts are leading to transformative findings and paving the way for future drug development, more clinical trials, and ultimately to treatments and cures. Our goal at the Hereditary Disease Foundation is to ensure that we continue to support these exceptional scientists and foster their collaborative efforts. We provide funding to promising young investigators who are just beginning their careers, as well as distinguished experts who have dedicated their life's work to studying HD. In August, we hosted our biennial scientific conference and brought together 250 researchers from around the world. For 4 days, they shared significant scientific findings, engaged in important dialogue, and forged critical partnerships that will continue to advance the field of HD research. There is a tremendous sense of hope, optimism, and momentum in the Huntington's disease community. The rapid advancements that are happening in science and technology can only be realized with the support of all of you, our donors, and true partners in research. Please join me in making a donation to the Hereditary Disease Foundation. It is with your generosity that we are able to continue funding the scientists we've met this evening and others who share their determination and passion. And your support provides hope to the many families impacted by Huntington's disease.
Hi, everyone. Thank you for joining us. Now it's time for Ask the Scientists. So please, I'd like to introduce Dr. Leslie Thompson, who is the chair of our Scientific Advisory Board. Leslie, please take it away.
Hi, Megan. Thank you. And thanks everyone for being here. It's really an exciting time. It was so fantastic to see all these young investigators who are sharing their passion in working with Huntington's disease. And it's also just wonderful to share this time and have this opportunity to talk about the exciting research happening in HD and to welcome so many members of the HD community. We have researchers and HD family members from all over the world joining us from countries such as South Korea, Turkey, Sweden, and New Zealand. You've already met our panelist, Dr. Rachel Harding, who is a driving force in the HDF's Young Investigators Forum. She's an incredible collaborator and really a generous person in her work and providing people with resources and reagents. And at the HD, she was dubbed the collaborator-in-chief. I don't know if you know that, Rachel, for all the research projects that she works on. And then Steve Finkbeiner, our recent Leslie Gary Prize winner for innovation in science. So he is the consummate collaborator, inventor, innovator, and he's spent his life really thinking about ways to overcome challenges and hurdles. And if he can't overcome it with what's in hand, he'll just invent it. And he's just an amazing researcher with a passion for treating this disease. So Steve, I think you had wanted to make a comment before the questions started, I believe.
Yeah, thanks, Leslie. It's such a pleasure to be here. And I also want to just welcome the whole audience. But I mostly wanted to say thank you to the Geary family, the Hereditary Disease Foundation, and especially Nancy Wexler, HDF's president, for her incredible support and friendship over the years. Many years ago, when I was a lowly postdoc laboring in isolation in a lab, she found me and critical to sort of recruiting me into the HD field. And I've been here ever since. So a huge note of thanks to everyone.
Yes, totally agree, Steve. And there's my picture of Nancy over here on the wall. And she did the same for me when I was a young postdoc. So let's start out with the questions. And maybe I'll direct this to you, Steve, from Susan. We know about the clinical trials that are underway, but when I hear the Bristol-Myers Squibb, and thanks you, George, is interested in developing drugs for HD, I ask if there's other drug companies working on HD that we don't know about. Are there a lot of companies? How does it work? At what point might it be publicly announced? Kind of what's the process for that?
Yeah, so it really varies by company. Some actually want to keep the work they do more private for a while. And so sometimes it doesn't become public until it reaches a certain stage. But others announce these programs and projects at an earlier stage. And yeah, I think you even heard on the video tonight, there is a lot of interest in Huntington's disease. I think as people have pointed out, one huge advantage for at least drug companies interested in studying neurodegenerative diseases is that they can be 100% confident that when they enroll a patient, they know that the cause of the disease is the same for every patient. And that's not always true for a lot of disorders. And it turns out that's very important in terms of knowing that when you test a drug that's really designed to target the mechanism of disease, that every patient who's enrolled in a trial has a realistic chance of potentially benefiting from that therapy. It increases significantly the chances of being able to show that a therapy is effective. So I'm aware of quite a few drug companies, both in stealth mode, but also publicly, that are working in this area. And really, I think one of the exciting things you heard tonight about sort of kind of a golden age of HD research and therapeutics, and I would echo that remark, is that people really have thought about a variety of strategies to target Huntington's disease. And I think I'm especially excited because increasingly these strategies are really rooted in good science. And so it gives me a greater hope that we're going to really see some things that are going to have a great impact on the disease.
Thanks, Steve. And Rachel, maybe what's your perspective on maybe some of the most promising or things you're excited about in HD research?
Well, I think there's, a huge number of really cool technologies. Some of the work that Steve's doing with AI is, that's such a big and emerging field. And it's being applied, not just looking at cells, but looking at molecules, understanding things at a very small, detailed level, all the way through to studies on people and other bits and pieces, helping detect symptoms and other kind of measures that we might be interested in. And that's just, exploding right now. And that's really exciting. And a lot of it is far beyond my understanding. But it's going to be applied for not just Huntington's disease, but other diseases. And I think that's going to be really, really exciting as well. I think it's interesting as well that we've got so many different companies trying both ASOs, but also these splicing modulators in the clinic. And I know that Roche announced recently at HSG that they are also interested in finding these splice modulators. So these are drugs that might be taken in a pill format, for example, or as a syrup that can lower the levels of the Huntington protein. And so it's really cool to see companies trying these new strategies and these different ways that they can target the Huntington protein.
Thanks, Rachel. And from Kathleen Farrell, a great presentation. Thank you. I have a question about the mRNA technology used to develop the COVID vaccine. Is there a role for this technology in the research and therapeutics for HD? Do either of you have thoughts about that? I would guess that there would be, that this could be applied in some way. Steve, do you have any thoughts? Fun, right?
It's not, yeah. I mean, so Huntington's disease, importantly, is not a infectious disease. And so we certainly wouldn't be developing a vaccine that would try to engage your immune system per se to fight Huntington's disease. But I will say that a lot of the underlying technology of being able to manipulate and target mRNA specifically, for sure, is being something that's harnessed in Huntington's disease strategies. The audience may be familiar with a Nobel-winning prize technology called CRISPR, which is sort of, you can think of sort of genetic scissors that can cut DNA in very specific ways to achieve different goals. For a long time, that was mainly focused at DNA, but now there are new CRISPR scissors that work just on RNA. And so people have been very interested in editing RNA, and it looks quite promising in terms of even for Huntington's disease.
Yep. Thank you very much. And Anonymous, well, thank you. This is so inspiring. What's the timeline you're thinking for available treatments? And I think we heard that question on the video as well, where it's always impossible, I think, to predict what the timeline is going to be, but I agree that we're all working as hard as possible and as quickly as possible. And these new technologies that are out there, you heard Julia mention iPS cells, the AI approaches, Rachel's work, with purifying these proteins and really understanding what's going on and having tools we've just never had before is really accelerating the pace of all this. But so as soon as possible would be an answer. So from Tacey Fox, hi Tacey, what a fabulous presentation. Wow. Dr. Finkbeiner, could you tell us a little more about how you're using machine learning to figure out what is going on in a neuron that has mutant Huntington, how you think that will expedite finding a treatment for HD? Also, using AI and machine learning to explore Huntington's role in DNA repair and how mutant Huntington makes the normal function ineffective. So basically, broadly, how you use machine learning and AI.
Yeah, so the short answer is yes. We're kind of throwing spaghetti at the wall right now. We've been so impressed by the power of these approaches and have been surprised to the upside in how they perform and some of the things they've enabled us to do. And on more than one occasion, these approaches have revealed things to our human eyes that were invisible before without those tools. And so it's kind of an exciting time for us. And I think that the key thing is that we lucked out a little bit. We had built these robots years ago for one particular reason, but one thing they're very good at is generating mountains of data. And that's exactly what machine learning and artificial intelligence needs to be successful. It's just large data that's actually oftentimes much larger than really the human brain can comprehend. But with all that data, machine learning approaches can oftentimes really hone in on some of the key important features. You saw one of the folks in my group, Julia Kay, talk a little bit during that video of ways she's used machine learning to be able to identify the key differences that we see in patient-derived neuron models of Huntington's disease based on stem cells. And the way that helps us is that when we find what the key differences are, even if they don't necessarily fit into neat and tidy bins that we scientists would love and wish sometimes that they would, those really key differences probably are the things that are Huntington is affecting the most. And we can use that to look for things that will fix Huntington's disease. And I think Lise Barbet, another person in my group, mentioned it tonight. Really look for things that'll make a sick cell back into a healthy cell, but in a really holistic way, so that we're not just changing one thing, we're really trying to get to the root of the problem and making those cells healthy. And we think that not only does that make a certain amount of sense, but we also think that if we can really get to the root of the problem, we're also going to hopefully find therapies that will have a much more stronger impact on symptoms and things like that when they're trialed in patients.
Thanks, Steve. So Rachel, I have a couple questions for you. So one of them, people were intrigued by that blue model you were holding.
Well, this guy.
There you go. I know, I have one of those. I love it. Can you talk a little bit about that? And then secondly, well, I'll get the next one in a minute. Go ahead and talk about that.
Sure. So I think this is the smallest version of this model that I have, which is the one that I currently have on hand. And this is actually from Ray Truant, who was featured in the film earlier. It's a 3D printed model of the Huntington protein molecule. So we now know where nearly every atom within the Huntington protein is. There are still bits that are missing in our model, but we have well over half of it now. And so the bits that we do know, we can build this special little shape. This is the shape of the Huntington protein. And then Ray 3D printed it for me and sent it to me in the post, I think. So yeah, this is nice. It's very helpful to, rather than thinking abstractly about what are all these molecules in our cells doing, but to physically look at the molecule sometimes is really helpful, at least to people like me who are really geeky about protein structure molecules anyway.
I 100% agree. I think it's incredibly helpful. So another one they were asking, saying, we've sort of heard about from Steve and Leslie how Nancy influenced their career, and asking, how did you get into HD, Rachel, HD research?
Yeah, so I mean, I was fortunate to be offered, you know, with my postdoctoral studies, the opportunity to start thinking about working in HD. And one of the first conferences I ever went to was a CHDI Therapeutics conference, where I met Marcy McDonald, who is also an extremely esteemed female HD scientist. And I remember her saying to me that once you get the HD bug, you will not be able to work on anything else. And she was absolutely correct. So yeah, I think the thing that's really interesting for me scientifically is understanding how this very small genetic change in the grand scheme of this huge Huntington gene can give rise to, you know, the phenotypes and the symptoms that we see in patients. But I think the thing that keeps me engaged in all of this is the community of, you know, the scientists, the clinicians, and the patients working within this space is just really inspiring and something I definitely want to keep doing.
Thanks, Rachel. Completely agree. So Steve, how are you able to develop your amazing inventions? Do you collaborate with tech people or invent them yourself? I kind of know the answer to this, but go ahead.
Yeah, no, this is great. Like I'm halfway through my written answer, so I can just read it to Susan. So what I'll say is my parents bought me probably 7 suitcases of Legos when I was a boy, and so I think it really began with playing with a lot of Legos and just building stuff and being creative. They also let me tear apart expensive toys they bought me to understand how they worked, and then gave me lots of opportunities to build things growing up. So I think that was a big part of it. I also, my one part of my family are farmers from North Dakota. When you're on the farm, you kind of have to fix things out in the field because you just can't get back in time. So you get to be pretty resourceful and creative. And then when I finally got to graduate school, I was in a physiology lab, and there's a strong tradition, at least in old school physiology labs, of building your own rig and, you know, basically writing your own software and things like that. So I think that really showed me that you can do it. And it's actually a lot of fun sometimes to sort of build stuff.
Cool,
but just to answer her question, we do also collaborate with lots of tech companies. And in fact, you know, a really catalytic phone call I got was from Google in 2015. They kind of cold called us and said that we generated enough data to be interesting. And so that started really a big step for us in terms of really trying at a much better, higher level of integrating artificial intelligence in what we do.
Thanks, Steve. So Rachel, wow, what an exciting, wonderful presentation. So what have been some of the most exciting moments in your research so far?
Oh, that's a tough question.
I know, that's a tough one. Huh?
I think one of the things I find the most exciting is when you run an experiment and then you feel like you're holding a piece of information that no one else in the world knows at that time, right? And that is when you make those kind of discoveries, that's really, really exciting. And I was fortunate earlier this year to present on some of the work we've been doing, understanding how Huntington is able to bind different types of genetic material. And that was at the HDF conference that happened earlier in August. And that was when we first started to get data on that project. That was really exciting. And it, you start instantly connecting all the dots in your head about how this might explain everything, but, obviously step by step. So I think that those for me are the things that I find the most exciting.
Thanks, Rachel. And I'll address this one. Kathleen Farrell says, do you receive funding from the HDF? I'm sorry, from the NIH. And we do. I'd say that US investigators for sure get funding from the NIH, but the issue is that sometimes it takes so long to have the application, you submit the application, it gets reviewed months and months later, then months before council and things like that. And so it takes a long time. And the incredible thing about HDF and the funding that we've all received is that it allows you to do things right away, to do things, that you just think would be potentially pivotal, would be exciting, and might move the field forward very quickly. So it's that combination of opportunities and funding, I think, that really makes a difference in HD. Another, I don't know, Steve, if you started typing the answer, but about talking about using cells from actual patients, and we can both kind of address that. Go ahead if you'd like to.
Sure. Yeah. No, in fact, Leslie and I have been longtime collaborators in this regard. We were part of the original, in fact, this is relevant to the last question, NIH-funded effort to develop induced pluripotent stem cells for Huntington's disease, the community and from patients. And so one of the things we did here at Gladstone was recruited a lot of HD patients. I would perform the neurologic exam on them, and then we would collect biosamples to make those induced pluripotent stem cells. And then Leslie and I, for years, have been using those cells, turning them into relevant brain cells for Huntington's disease, medium spiny neurons, cortical neurons, different types of immune cells, and then using them to do a number of things. One is to initially just try to see if those cells behave differently than cells from other family members without the HD gene, or using CRISPR, trying to correct the genetic mutation to see if those cells, again, any changes or differences that we see in the Huntington's disease cells, whether those are corrected. And so that's very useful for helping us understand Huntington's disease, the mechanisms, and being able to use it as a tool to dig deeper. But it's also really useful as a platform for finding therapies. And so that's something Leslie and I have also been working on, especially trying to find those other genes in the genome besides the Huntington gene that influence how severe someone's Huntington's is. And so we've been working on a project together now, supported by the Hereditary Disease Foundation, to explore those with the idea that some of those might actually be therapeutic targets themselves for HD.
Thanks, Steve. So from Dominique Donaldson, thank you for the incredible presentation. Congratulations to all the contributors. How do scientists recruit volunteers with Huntington's disease for experiments and research? And I'll go ahead and do that one is I think, the thing that's so unusual about the HD field is the community, as Rachel was mentioning too, that there's this amazing community that works together that includes the families. And so the families are so engaged with the researchers and are so willing to work together with the scientists to advance science. So for instance, the iPS cells, the stem cells we were just talking about, the human cells that Steve was talking about, when we began those studies and getting the skin samples from individuals, that was, the community just came forward so willingly and so and wanting to participate in that research. So I think it's both through the local communities that we engage with the larger communities, with Enroll HD, which is a really important resource across the globe, with HSG, with other types of organizations that work very closely with the scientists. It's a very unusual community, I think. Steve, notice that you have both an MD and PhD. Did you initially want to be a neurologist? I guess you are.
Yeah, so I went to Yale, and they had a long tradition there of having the medical students do research. But my folks, I was adopted, and my folks hadn't gone to college. So they, I was a smart kid, and they thought a smart kid should be a doctor. So that's kind of the simple boneheaded approach of how I ended up in medical school. But then I found out that I loved research so much that after the first year there, I enrolled in what's called the Medical Scientist Training Program, which is the combined MD PhD program. And so got the degrees together, and knew that neuroscience was something that I really loved. And so neurology was really attractive and appealing to me.
Thanks, Steve. I think there's quite a few questions here about clinical trials and, what have we learned from the past that can guide us more effectively in the next stage of clinical trials? And then also from people, what can we be doing, families be doing now while waiting for clinical trials? Rachel or Steve, if you'd like to talk about that a little bit, and then I can also join in.
Yeah, I saw the question about, what can folks do rather than just waiting. And there's all sorts of ways that the patient community can be involved, as much or as little as you like, in HD research, but your help and contributions is really what is driving things forward. And in particular, people like the HDSA have their trial finder. So there's all kinds of studies that you can get involved in with like that. Not all of them are testing drugs. Some of them are just like helping us understand how to track Huntington's disease. And so there's things like HD Clarity, which Lauren Byrne, who was featured earlier, is involved with Ed Wild, looking at spinal fluid samples. And, you know, that's really, really important. There's Enroll HD, all these other studies, which just provide this huge wealth of data that then very clever scientists can look at and try and understand the patterns and what's going on and identify things like biomarkers, which I think is also mentioned in another question.
Yeah, thanks. Yeah, and I think we're learning so much from the clinical trials, and even, you know, the ones that are quote unquote failing or maybe have been stopped. We're finding out so much information from that, and like you said, there's the materials now for the first time we can use to study biomarkers and what changes with disease. There's another question about you all talk about collaboration a lot. Can you talk about more what that means, and do you all get along? Laughed all out. Yeah, I think collaboration is just critical, has been from day one of trying to find the HD disease gene, has been an essential part of HD research. It's, you know, it's a more rare disease, it's a devastating disease. It's something that you've heard in the talks that we have to really work together to solve this. From Judith Friedman was talking about this, that we all have our areas of expertise, and you want to bring those things together and work together. And it's a very collegial, collaborative field. I think most of us feel like it's our second family in many ways. The people, you know, we on this call, we've all known each other for a very long time and been colleagues. I don't know if you all wanted to mention anything too.
Yeah, I would just add that, you know, collaboration is, you know, it's good and it's fun and it's great to have that sense of community, but it's also what gets science done faster. I think, you know, I think Lauren said it earlier in the video, which is like, you can't be, the days of being, you know, a single scientist on your own in a lab are long over. Like, we have to work together collaboratively. And, you know, we've just been through this terrible pandemic, which is still really going on, but you know, that's a great example of what can happen when a global community of scientists come together. We now have this plethora of vaccines, these amazing antiviral drugs from Pfizer and other companies coming through and really helping people. And, you know, it'd be amazing if we could apply some of those principles to HD research, working together openly and collaboratively. And I think it's already happening, but, you know, HDF is encouraging that to the next level.
Thanks,
yeah, I'll just say, because I think this is really where HDF shines, is that they have been, so you heard reasons why collaboration is critical, you heard, you know, some positive things about it, but HDF's really been catalytic in this regard. I think they, from the very beginning, both with the format of their meetings and the kind of culture they created, I think, in my opinion, because I work in other fields as well, really stand out. They have played an absolutely direct role in creating a model community, in my mind, of people who are really encouraged to collaborate, and has really fostered and that sort of flame, I think, that a lot of scientists feel, and I think also just their commitment to young scientists too, and getting them at an early age, you know, I think has really helped contribute to that. So we owe a huge thanks to HDF. They've really set the tone, and they're a major mechanism for collaboration.
Yeah, thanks, Steve. I couldn't agree more. And just to say, there are more questions in the Q&A, and we will get those answered. And some of them go directly to people that are working on it, or clinicians who might be able to answer some of those questions that are there. And echo that HDF and Nancy, and it's been pivotal in this field and bringing us together. Megan, thank you very much.
Thank you, everyone. And I'm so happy that we ended on the theme of collaboration, because you all touched on it so well, and everyone in the video touched on it. And it's just so important, not only the collaboration between the scientists themselves and their work, but the collaboration between the families and the scientists, and even between the families themselves and supporting one another. Coming from an HD family myself, I just find that when I meet other people who also come from HD families, you form this just really strong bond right away, because you just understand what people are going through. So I'm really glad. Thank you everybody for all of your questions. They were all terrific. Thanks for being such an engaging audience. Thank you, Leslie, Steve, and Rachel for sharing your expertise with us and answering our questions. And I'd also like to thank our Gala sponsors who will be listed after this, you, our donors, and everyone else for joining us. And thank you to the researchers who presented this evening. We're so excited about all of the work. It is your research that gives all of us great hope. Thank you, everybody, for joining us.
Thank you. Bye.